Welcome back. In our last lecture, we covered enzyme basics, classification, and coenzymes. Now we explore what controls enzyme activity in real systems. Why do some enzymes run fast and others slow? Why does fever change metabolism, or digestive enzymes work only in certain gut regions? Let's ask the big questions. First, enzyme activity means how quickly an enzyme converts substrate into product. It is measured as reaction rate per unit time under defined conditions. Activity depends on the enzyme structure and environment. Small changes in conditions can raise rates dramatically or halt reactions almost completely. This topic is central to exams and clinical practice because it explains drug actions, fever effects, and digestive function. We'll unpack each factor one by one. Let's begin with temperature, one of the most intuitive yet important influences on enzyme activity and stability. Temperature changes molecular motion. Warmer conditions increase collision frequency between enzyme and substrate, speeding reaction rates up to a limit. Each enzyme has an optimum temperature where activity peaks. Beyond that, structural bonds break and the enzyme denatures, losing function rapidly. Denaturation means the enzyme's three-dimensional shape unravels, the active site deforms and substrate can no longer bind productively. Clinically, fever raises body temperature and can alter metabolic rates. Mild increases may speed some reactions, but severe fever risks enzyme dysfunction. A temperature activity chart helps visualize this. Color-coded zones show increased activity, optimum, and rapid decline with denaturation. In labs, we control temperature precisely with water baths or thermal cyclers to keep enzymes functional and reproducible. pH changes alter the ionization of amino acids within the active site, shifting charges that are critical for substrate binding and catalysis. Every enzyme has an optimum pH. For example, pepsin in the stomach works best at low pH, while trypsin prefers the small intestine's neutral to slightly alkaline pH. Extreme pH levels can disrupt hydrogen bonds and ionic interactions, causing active site distortion and loss of activity, similar to thermal denaturation. A pH activity graph shows a bell-shaped curve. The peak represents the optimum pH where ionization states favor catalysis and binding. Digestive enzymes are optimized for their compartments. Acidic stomach enzymes digest proteins, while alkaline pancreatic enzymes act in the small intestine. Clinically, antacids and acid blockers change stomach pH and can influence enzyme activity and drug absorption, so pH matters in therapy. Substrate concentration influences rate. At low substrate, rate rises with more substrate. At high substrate, the enzyme becomes saturated and rate plateaus. The plateau defines Vmax, the maximum rate when every enzyme active site is occupied. Km indicates substrate concentration at half Vmax. Enzyme concentration also matters. More enzyme molecules increase maximum achievable rate, assuming substrate is available in excess. Competitive inhibitors bind the active site, blocking substrate. Increasing substrate concentration can overcome competitive inhibition in many cases. Non-competitive inhibitors bind elsewhere, changing enzyme shape and reducing activity, regardless of substrate concentration. This lowers Vmax but not Km. Many drugs work by enzyme inhibition. Examples include ACE inhibitors and statins. Understanding inhibition helps design safer, targeted therapies. In the lab, enzyme activity is measured by tracking product formation or substrate disappearance over time using spectrophotometry or linked assays. Color-coded charts are useful teaching tools. They let viewers see how activity changes with temperature, pH, and concentration at a glance. Animated diagrams reinforce concepts, show the active site, substrate binding, and how pH or heat distort the active site and reduce activity. Practical tips: store enzymes cold, avoid repeated freeze-thaw cycles, monitor patient temperature in metabolic disorders, and consider drug interactions carefully. Quick review: activity equals reaction rate temperature, and pH have optima concentration, and inhibitors change rates. Clinical and therapeutic implications are significant. That concludes Enzymes Part 2. Review the diagrams, remember the charts, and apply these principles clinically and in the lab.